And on the session control, we'll move to the channel side to Dr. Jichuan Xiong.
Okay, thank you. Yes. Okay, I'm going to share myself. My name is Jichuan Xiong from Nanjing University of Science and Technology. I guess Glasgow is looking forward to dinner. We are looking forward to bed. I'll make it short. First, I'll give an introduction of our lab, our team, and then I will show a video of our lab. I guess most of the friends in Glasgow already know where we are, but we still have some audience who is not familiar with our university. Nanjing is a city which is very close to Shanghai, and Shanghai everybody knows. This is our campus, very beautiful in different seasons. This is our team. Our team actually we can divide it into a few subgroups in computation, in optical imaging, in photographic imaging, and application development. I introduce one by one. The facilities of our lab, you can see a few pictures we take in the lab. The facilities of the group, we have two labs with about 300 square meters clean room, one computation cluster, and electronic, mechanic, and bio workshops. We also have some shared big facilities like SEM, AFM, with other groups, and the capabilities of this group we can do normally in we can do fluorescent, convoke imaging, super resolution, optical imaging. Also, we do have systems for wafer inspection, terahertz imaging, wavefront sensing of optical systems, and also we do scientific computation. These are some pictures of the main facilities. For the research of this group, mainly we are supported by the Ministry of Science and Technology, the Natural Science Foundation of China. Also, we are supported by our international collaborators, especially Glasgow. The team is led by Professor Xuefeng Liu, a member of national experts. We have team members focused on optics, photoacoustic, biomedical, and instrumentation. Of course, since we are doing instrumentation, we are also doing commercialization of our instruments. I'll introduce two major directions of our research. One is on optical imaging. Optical imaging, we are heading for super resolution imaging. Mainly, one of the big applications is for in situ analysis of biological structural characteristics. The initialization of this research is actually is trying to understanding to imaging and to understanding the fundamental of life process. As everybody know, for imaging techniques, what we are dealing with now with optical imaging is above the subdiffraction, above the diffraction limit, which is a few hundred nanometers. And there are still short of optical tools in a range of 100 nanometer to tens of nanometers, which is very important for cell development, for the study of cell development, study the gene conservation, and also to study the dynamics and conformation of macro molecules. So the situation for now is that there are techniques for imaging this process. There are non-optical images. There are also some other imaging methods, which is all has different kinds of limitations. For optical super resolution, there are already quite a few techniques which is well established, but each of them has some certain limitations, and we proposed a new technique which is called PIMI, and we published quite some paper on this, and now we are applying this technique toward in applications. The starting point is that in optical imaging, if you are familiar with the optical imaging system, you will heard a lot of point spread function, not in different kinds of imaging techniques. In point spread function, the diffraction limited optical imaging systems are always limited by the point spread function, but you can still get information inside the point spread function. And what we are aiming for is to get information within the point spread function. By doing this, we can solve a few questions which we are facing now in optical imaging systems. One is that our method is that we in an image system we can manipulate the incident of the light state. We can manipulate the photon state of the illumination light, and in the sensing system we demodulate the photon state, and by filtering and demodulation we can break the diffraction limit. And in the right figure, you can see a comparison of conventional imaging system and our imaging results. The detailed structures in a seal can be clearly visualized with our method. The principle is mainly based on microscopy optical path, but in a conventional microscopy optical path, we add our modules to manipulate the photon state of the illumination, and in the sensing imaging, in the sensing system, we do the photon state demodulation and fitting and filtering to extract the parameters of the photon state. With these parametric images, we can resolve details which was not obtained in conventional microscopy. Here is a seal, an image of a seal, and also we imaged a lot of nanoparticles. In the right figure, you can see the image of a gold nanoparticle with diameter of 100 nanometer. This is the image results of the seal. You can see the detailed structure of subcellular structures. Here are some imaging results also of seal and nanopores, gold nanoparticles, and DNA nanoballs. Based on this technique, we are making some attempts to visualize the passage network of BMN. This is collaborated with BIT. Another application which we are making efforts now is to apply this method for viral detection. For viral detection, we use a gold nanoparticle. We connect the viral particle to the gold nanoparticle, and in the imaging system, we can make use of the plasmonic diffraction of the gold nanoparticle. When the gold nanoparticle was connected with a virus particle, the diffraction symmetry will be broken. Then we can use this broken of symmetry to sensing the nanoparticle, to sensing the viral particle. Also, we are doing research on photoacoustic imaging. In photoacoustic imaging, we are trying to combine optical imaging with photographic imaging to make a multi-modality system for optical imaging and photographic imaging. And this is a prototype we developed. We made some visualization of propagation of photographic wavefront in a phantom. Of course, we are also trying to commercialize our instrument. This is a slide showing a few instruments we have already developed. Based on this instrument, we can obtain different kinds of diagnosis data, and we transfer this data to cloud computation. And cloud computation is supported by AI and also our collaborators of clinic doctors, and also based on the Chinese traditional medicine theory to make personalized diagnosis. Of course, we have long-term plan together with our collaborators like CTU of Hong Kong to develop this kind of instrument and algorithm and database to a platform which can provide portable, wearable, and personalized diagnosis instrument and platform for disease. And AI, of course, everybody uses AI. In our method, AI is for imaging denoising, signature extraction, and diagnosis. Our collaborators, like Professor Dong, who has already given a talk, and some hospitals. Of course, we would like to mention our funding bodies and also our collaborators our international collaborators, especially University of Glasgow. Also, our team members, our students who has already worked very hard these times from this year and also these days, and thank you to them. Okay, that's my talk. Any questions?
Oh, thank you very much. If you have any question.
Oh, I think I forgot one thing. We have a video. You can have a very quick look of our lab. I can show it quickly. Yeah.
Okay, you can show.
Okay, I'll forward a little bit, then you can skip the beginning. This is my colleague, Dr. Xu, who is introducing our lab. I'll go directly to the lab. This is a hallway with all the information of our group, and we are going to the lab from here. We have two labs with clean room. All the optical measurements and systems are in the clean room. Here are some systems developed by us, which some of them are already commercialized. This is a wafer inspection, which I mentioned. And also we have some facilities owned by our own group, like confocal imaging it's a facility with a femtosecond laser, which can do full photon microscopy, and also for bioimaging. Here are some systems developed by our own, by our group. The multimodality optical imaging and photographic imaging system, and also another prototype on this based on this technique. Here is our PhD student Gu Kai. Our another PhD student Jin Xiao, who is introducing our optical super resolution imaging system. This system can also do fluorescent super resolution imaging. Here is another lab, mainly with terahertz imaging system. Here, this is our terahertz imaging system, and some commercial instrument for wavefront sensing of complex optical systems. Of course, coffee room. I will not introduce too much here. Okay. Thank you.
Okay, thank you very much. I have a question for your commercialization machine, higher resolution machine. How much you will sell for each machine?
Which one?
High super you build
Super resolution optical imaging system. That one, if we only count the cost, not real commercial price, I think we can make it to under 500K RMB.
Okay.
Can you hear me?
Yeah. Thank you very much.
If you want to buy one, we have friend price.